importance of Th9 and Th17 in physiology and disease. So, Dr. Goswami, please.
Thank you very much, Dr. Pata. Am I audible?
Yeah.
Yes.
Yeah. So I need to share the screen again, I believe.
Yeah, that's fine.
So is the full screen coming?
Yes.
Yes.
Okay,
Yes.
great. Great. So I would like to thank the Society of Information Research, Dr. Giri, Dr. Chandrasekhara for extending importance of Th9 and Th17 cells in physiology and diseases. Even though I work a little bit on Th9 cells, not so much on Th17 cells, but I will try to, in this half an hour, I will try to give a glimpse of the importance of these cells. Little bit of the work which we started, which I actually started during my PhD, and where we are in terms of the regulation of Th9 cells and the importance of Th17 cells and physiology and diseases. With respect to T helper cell development, it is very well known that it is not an outcome of one single signal. It is an outcome of multiple signals. And when an antigen presenting cell is presenting and processed peptide in the context of class 2 MHC molecule to a naive CD4 T cell, which has the TCR to identify that particular peptide, this interaction in the presence of co-stimulatory molecules can give rise to effector T helper cells, which are very critical for providing robust immune responses against variety of pathogens. And in the process, what these effector T helper cells do, they not only protect us from intracellular organisms, they also help us in fighting against extracellular parasites, fungal infections, providing help to B cells. And when the infection has been taken care of, when there is no further requirement of inflammatory assault to induce the tolerance. So this is a very simple schematic of understanding what T helper cell development is all about. But when we move to see what are the signals which are required for these particular purposes, it gives us to the T helper subset differentiation. And there are additional parameters apart from the peptide MHC molecule T cell receptor interaction, apart from the co-stimulatory interactions. There are various polarizing cytokines which can actually lead to the differentiation of these naive T helper cells into distinct T helper cell subsets. They are also aided by various transcription factors which are expressed, and this gives rise to this bunch of T helper cell subsets. It started off with Th1 and Th2, now it has expanded, and this is also probably not the exhaustive list of T helper cells which we can see. There are additional subsets which we are really not going to discuss today. And each of these T helper subsets are characterized by the secretion of signature cytokines. So for our interest, we will be focusing on Th9 and Th17 cells. And as the name suggests, Th9 cells secrete huge amount of the cytokine interleukin 9. And Th17 cells secrete not only interleukin 17, but they also secrete interleukin 21, interleukin 22, interleukin 26, and GM-CSF as their key cytokines. So in that context, Th17 cells is a little bit more complex. Th9 cells also express some amount of interleukin 10 and interleukin 21, but their levels are definitely lower than what a Th17 cells would express, or maybe what a Treg cells will secrete. The other thing to notice is that some of these transcription factors are shared across these different T helper subsets, as well as the polarizing cytokines. So for Th9 cell differentiation, we need the signals interleukin 4 and TGF-beta. For Th17, the common signal is TGF-beta. And in the presence of TGF-beta only, naive T cells give rise to regulatory T cells. So if we actually take a look into the timeline of major T helper cell discoveries, we can figure out that there are probably the discovery of very important T helper subsets within a frame of 10 years or so. So the seminal work done by Mossman and Hoffman back in 1986 paved the discovery of T helper cell subsets with the Th1 and Th2. For a long time, for about 10 years, this was the only established thought that there would be either Th1 cells or there would be Th2 cells. Th1 cells will be fighting off against intracellular pathogens. Th2 cells will be fighting off against extracellular parasites. Now, if we move 10 years from their discovery, in 1995, the first report of regulatory T cells came into the picture. And the discovery of our topic of interest, Th17 cells, came in 2005. So again, another 10 years. But Th9 cells, it came 3-4 years after the establishment of Th17 cells. So back in 2008, this is when Th9 cells were actually established, or the concept of IL-9 secreting T helper cells came into the picture. In the bottom of this slide, you can appreciate the key transcription factors, or if you would like to call them as the master transcription factors of distinct T helper cell subsets and their discovery. So GATA3, which is a Th2 specific regulator, got discovered in 1997. 3 years down the line, Th1's Tbet was discovered, and FOXP3 was found to be very critical another 3 years from the line. So this gives you an idea not only that there has been an increase in the different T helper cell subset family members, but it also adds up to the complexities. And to add up to the complexity, we need to understand why there are these complexities and why these complexities are critical in maintaining a proper healthy physiology, or if that physiology is not able to be taken care of, what is the reason that is going to lead to different diseases. So as I mentioned, Th9 cells, I'll start with Th9 cells, even though it got discovered a little later than Th17 cells. The main cytokine which is produced by Th9 cells is interleukin-9. And if you like to see that the source of interleukin-9 is not only Th9 cells, there are other cell types which can also produce interleukin-9 in different levels. For example, innate lymphoid cells, they can secrete interleukin-9 when they are when they're infected with a particular parasite, or maybe during an allergic inflammation model, as example by Papin model of allergic inflammation. In vitro ILC2s, if they're stimulated with interleukin-2, they can also secrete interleukin-9. Mast cells, they also secrete interleukin-9 in the presence of an allergic inflammation. So are NK cells. The cytotoxic T cells, CD8, they can also secrete interleukin-9 at some level, and we call these cells as TC9 cells. But apart from the T helper cell 9 family, there are also additional T helper cell members which secrete some level of interleukin-9 and if we compare that level of interleukin-9 with what the Th9 cells secrete, that level is less. So for example, Th2 cells, they can actually secrete interleukin-9, and interleukin-9 was originally defined actually as a Th2 cytokine because its expression correlated with the expansion of antigen-specific Th2 cell population. Th17 cells also secrete a little bit of interleukin-9, so are the regulatory T cells. But the physiology of the production of interleukin-9 from these cells are still not completely understood. Even though interleukin-9 can be produced by multiple cell types, interleukin-9 is also a pleiotropic cytokine. So that basically means it can act on multiple cell types. As this figure actually says, that it can act on lymphocytes as a growth factor for T cells. It can act on mast cells, again, a growth factor and survival signal for mast cells. It also enhances the protease and cytokine expression from mast cells. It can also act on non-hematopoietic cells like the epithelial cells or the smooth muscle cells, where they can activate the production of different chemokines. In epithelial cells, it can also induce goblet cell metaplasia. It also helps in the colony formation of hematopoietic progenitor cells as well. So our quest to understand why two distinct cytokines, namely interleukin-4 and TGF-beta, would result in an IL-9 secreting T cell phenotype. And this phenotype would be different or unique from the effect of these two cytokines individually. As you can see, in the presence of interleukin-4, we can see the differentiation of Th2 cells. In the presence of TGF-beta only, we can see the differentiation of regulatory T cells. But when we add IL-4 and TGF-beta together, we can see a huge level of interleukin-9 being produced. So our initial goal was to figure out what are the transcription factors which are downstream of each of the cytokine signal that would contribute to the expression of interleukin-9. And one of the transcription factors which we actually looked was the transcription factor Q1. And the rationale was very simple. Q1 was known to inhibit Th2 cell production. The cytokine TGF-beta was also going to suppress Th2 cell production. So we hypothesized maybe this transcription factor Q1 is probably acting downstream of TGF-beta signal. And so we took the help of the Q1 knockout animals. So the gene name for Q1 is SFPI1. And so in this knockout animals, the Q1 was actually knocked out in T cells. So we cultured them under Th9 differentiating conditions. And you can see from all different assays that the level of IL-9 at either protein or by the mRNA level, it was significantly impaired in the absence of Q1. So it suggested that Q1 is probably an important parameter or a transcription factor which is going to regulate the differentiation of Th9 cells. But when we looked into the interleukin-9 gene, we also felt it is not only going to be the transcription factors which are going to be important, but also the interleukin-9 gene can be regulated by epigenetic modifications. So we treated these Th9 cells with a known HDAC inhibitor, trichostatin A, and we would our expectation was probably that treating with an HDAC inhibitor would open up the interleukin-9 gene chromatin. And we could actually see an increased level of interleukin-9 in the presence of TSA, as well as an increased expression of interleukin-9 and Q1 when Th9 cells were treated with TSA. More importantly, this effect was dependent on the transcription factor Q1, because even though they were treated with TSA showed an increased level of interleukin-9, that effect was completely impaired when a P1 knockout Th9 cells were treated with trichostatin A. So we then wanted to also determine if there were additional parameters which P1 is regulating to control the expression of interleukin-9 gene in Th9 cells. And what we found was that one of the histone modifying enzyme, GCN5, was impaired, its recruitment was impaired in the absence of P1. So we actually had done chromatin immunoprecipitation experiments, and the x-axis we can see there are a couple of conserved non-coding sequences of interleukin-9 gene which were mentioned here. And we could see that GCN5's recruitment was impaired in P1 deficient animals. We also found that there were a couple of additional histone modifying enzymes which were differentially regulated in the absence of P1. but we only focused on GCN5. I will just explain it in the next slide why. Along with an impaired recruitment of GCN5, we also saw there were specific histone acetylation marks which were also impaired in the absence of P1. And these were some of the histone acetylation marks which are actually mediated by the GCN5. So not only in the absence of P1, there is a decreased recruitment of GCN5 to the IL-9 gene, but also that correlated with impaired histone acetylation, specific histone acetylation marks at the IL-9 gene as well. So we finally asked whether this histone modifying enzyme GCN5 would associate with P1, and we could see that using DNA affinity precipitation assay that indeed in the absence of P1, the GCN5 cannot work. So GCN5 associates with P1. This was not observed with the other histone modifying enzymes. We could not find out an association of the other histone modifying enzymes with the transcription factor P1. And we also went ahead to check that which domain of P1 was important for this interaction with GCN5, and we figured out that it was actually the activation domain of P1, which allowed the recruitment of So we then knocked out GCN5 by using siRNA, and that led to significantly impaired GCN5 promoter and IL-9 gene expression, as well as reduced specific histone acetylation marks, as you can see in the IL-9 promoter, which is shown by shown over here. So if I have to summarize this story, and this is something we started back in 2008. So based on this part of the story, we could actually say that downstream of TGF-beta signal was the transcription factor Q1, which was going to bind to the interleukin-9 gene. And there was also the interaction of the Q1 with the histone modifying enzyme GCN5 to regulate this process. For the sake of time, I'm just going to very briefly, in the same time, we also figured out that interleukin-4 STAT6 signal was also important for Th9 cells, because downstream of STAT6, the transcription factor IRF4 was also important, as well as the STAT6 signal was able to suppress the expression of FOXP3, which was induced by the TGF-beta signal. STAT6 also suppressed the expression of Tbet, which is also the master regulator of Th1 cells. So a fine balance of interleukin-4 and TGF-beta signal was required for the optimal expression of interleukin-9. So this is the summary of the work which was done in 2010. And 5 years down the line, if we actually look into the transcription factor network of Th9 cells, we could see a huge increase. We could see that not only the IL-4 TGF-beta signaling is there, you could see that TGF-beta family member activin A also positively regulates this IL-9 gene. We could see that interleukin-2 STAT5 signaling is also important for the transcription of interleukin-9 gene, as well as there are additional cytokines like TSLP, interleukin-33, interleukin-25, which would activate either the STAT5 or the NF-kappa B transcription factors to regulate the transcription of the interleukin-9 gene. What is important and what people have always asked is, since we could not find a single transcription factor which is absolutely necessary for Th9 cell development, whether we can call Th9 cells as a specific T helper cell family member. I can answer that question towards the end, but I would like to move on and give you the relatively newer data or the complexity of the Th9 cell transcription factor network. And I'm not going to talk a lot of these things. I'll just only mention that if we look into the bottom at the nucleus, what are the transcription factors and co-activator molecules which are relevant? So we can see that this MAT signaling, which is downstream of TGF beta, is important, as well as the transcription factor BATF, the transcription factor FOXO1. This work has been done by Amit, and his also work on HIF if 1 alpha is also very relevant in this case as well. We can see that co-activator P300 is also important for the regulation of interleukin-9 gene. So if a cytokine is regulated in this fashion, in this complex network, it begs to the question, so what is the use of this particular T helper cells? But the story does not stop there. So apart from the cytokine, the transcription factors, the epigenetic modifiers, we also wanted to see what are the additional parameters or what are the additional factors which are regulating this interleukin-9 gene. And we looked into the role of the micronutrients, mostly the vitamins in this process. And one of the vitamins which we looked at was the vitamin D, the fat soluble vitamin D, or the biological active form is also known as calcitriol. So some of these data slides will have the word calcitriol. So on the top of this figure, you could see that we started off by mentioning there are 2 conserved non-coding sequences of interleukin-9 gene, but now it has been expanded. So CNS1 is basically the interleukin-9 promoter. CNS0, which is roughly 6 kilobit base pair upstream, is the 2 major CNSs which I had described a while back. But then there was also another CNS which is identified roughly 25 kb upstream of the transcription storage site. And so what we saw initially was that calcitriol would downregulate the expression of IL-9 gene from Th9 cells. So we wanted to check whether the transcription factor recruitment was altered in this IL-9 gene in the presence of calcitriol. And we could see here that in the presence of calcitriol, there was a significantly reduced recruitment of PU1, mostly at the CNS1 or the IL-9 promoter. And additional experiments using cell lines and primary T cells, we could actually see that PU1 would interact with the vitamin D receptor, which is one of the receptors for the signaling of vitamin D. So it gave us an idea that maybe PU1, when Th9 cells are treated with calcitriol, VDR is interacting with the transcription factor P1, and it is not allowing that transcription factor to bind to the interleukin-9 gene, most specifically to the interleukin-9 promoter, and thereby suppressing the regulation or the expression of interleukin-9 gene. We also saw whether there was an correlation or a role of the Treg cell-mediated transcription factors. The reason behind this idea was, since TGF-beta was the common cytokine for the differentiation of Th9 cells and Treg cells, we wanted to check what actually happens if Th9 cells were treated with calcitriol in terms of the expression of FOXP3. So Th9 cells, when treated with calcitriol, not only increased the expression of FOXP3, but it also increased the recruitment of FOXP3 to the interleukin-9 promoter, as you can see from the chromatin immunoprecipitation experiment. So this tells us there are 2 things happening. One is not only the positive regulators of Th9 cells, their expression and recruitment is getting impaired in the presence of calcitriol, but calcitriol also increases the recruitment of the negative regulator of Th9 cells, such as FOXP3. As I had shown previously that interleukin-9 gene can be regulated epigenetically, we also looked at whether calcitriol was actually regulating the epigenetic modifiers differentially in the Th9 cells. And we could see that histone deacetylase 1, this particular enzyme was significantly increased recruitment was found in the presence of calcitriol at all the 3 CNS of interleukin-9 gene. However, if we try to reverse the effect of HDAC1 by treating the cells with TSA, we could see a rescue effect of interleukin-9 gene, which was shown, which is kind of shown here by looking into the flow cytometry data and also the ELISA and the qPCR data. Additionally, Th9 cells, if they're treated with calcitriol, we could see the downregulation of recruitment of some of the specific histone acetyl transferases to the interleukin-9 gene promoter more specifically. So lysine 9, lysine 14 histone 3 acetylation, and lysine 5 histone 4 acetylation, all were actually impaired in Th9 cells treated with calcitriol at the interleukin-1 promoter more specifically. So what is the purpose of so tight and complex regulation of these Th9 cells? So Th9 cells have been shown to be involved in allergic inflammation. And so it does not play a beneficial role, it plays the bad role in allergic inflammation. It also, actually the interleukin-9, which is secreted by Th9 cells, along with the amplifier cytokines, interleukin-25, interleukin-33, TSLP, they can act on different cell types like mast cells to activate these mast cells and secrete their mediators. It can increase, these cytokines can increase the immunoglobulin E production from these cells. It can lead to mucus and chemokine production from epithelial cells, and also can enhance the eosinophil mediated survival, infiltration, and mediator secretion. So altogether, these Th9 cells are responsible for chronic allergic airway diseases, food allergy, and allergic skin inflammation. So what is the good role of Th9 cells? We'll not have time to discuss, but it helps in preventing helminthic infection. So Th9 cells have been shown very clearly to expel these worms from our body very efficiently. The role of Th9 cells in cancer is a little bit complicated because Th9 cells have been shown to play both a good role and a bad role in different types of cancer. So in promoting tumors, interleukin-9 can actually either increase the number of regulatory T cells in certain cancers, mostly the lymphomas and hepatocellular carcinomas. It can also increase some of the anti-inflammatory mast cells activity. So it also promotes the proliferation of the tumor cells and inhibits the apoptosis of tumor cells. So this is the bad role which Th9 cells are doing. But on the other hand, in different models of melanoma, it has been found that Th9 cells are recruiting, basically they are increasing the lymphocyte infiltration in tumors, they are enhancing mast cell activity, they are promoting the survival of dendritic cells, and also enhancing the killing of natural killer cells to eliminate the tumor. So if we were to strategize to target the Th9 cells in allergy and cancer, if we are targeting IL-9 or IL-9 receptor or the epigenetic regulation of interleukin-9 gene, these targets can specifically look for amelioration of the chronic allergy asthma, or maybe amelioration of certain cancers, mostly the lymphoma. But on the other side, if Th9 cells are going to be used as a cellular therapy against antigen-specific Th9 cells, that can be done in melanoma patients. But this requires further studies. With respect to allergy, I would like to mention that there was a drug which was a monoclonal antibody against interleukin-9, which was developed by MedImmune back in 2010. And when it was administered to patients having mild to moderate asthma, it showed a favorable safety profile, and it led to a potential reduction in the asthma exacerbation. But when the results from a larger phase 2 study came, which also involved moderate to severe asthma patients, it actually showed not any better improvement or improving clinical activities. So it shows that targeting interleukin-9 in maybe a selected subgroup of patients with asthma or combining the interleukin-9 blockade with any other anti-cytokine regimens might provide better outcome. So I don't know how much time we have, so I can carry on accordingly.
You can go on another 5-10 minutes, no issues.
Okay, sure. Okay.
Yeah.
So I will just try to finish. So from Th9 cells, I'm going to change the gear to Th17 cells. And again, the reason is very simple. This change is very easy to make because of the cytokine TGF-beta. So for the polarization of Th17 cells, you can see that there are plenty of cytokines which are required. The most common cytokines or the most important cytokines are interleukin-6 and TGF-beta. The cytokines interleukin-21 can replace interleukin-6, and interleukin-23 is required for the maintenance of these T helper cell subsets. As I have mentioned, these T helper cell subsets also secrete various cytokines. Interleukin-17. Interleukin-17 has, at least there are two family members, interleukin-17A and 17F, which are secreted by Th17 cells. Also interleukin-22. It is also secreted by T helper 17 cells. And T helper 17 cells have been shown to be working on different type of immune cells and non-immune cells as well. So depending on what cytokines we are looking, which are secreted by Th17 cells, it will result in the activity or function of various immune and non-immune cells. For example, if we are looking into interleukin-17A or F, it can act not only on macrophages, it can recruit the neutrophils to the site of infection, it can act also on epithelial cells, it can act on fibroblasts. Interleukin-22 is important because interleukin-22 is actually going to be critical because of the transcription factor aryl hydrocarbon receptor, AHR. And AHR induces interleukin-22 with ROR gamma T, the master regulator of Th17 cells. Interleukin-21, it can act on CD8 cells, it can act on natural killer cells, and the cytokines which promotes interleukin-21 expression is IL-6, TGF-beta, and inducible co-stimulatory molecule. And these guys can actually induce the transcription factor CMAF. So for interleukin-22, the transcription factor AHR is important. For interleukin-21 expression, the transcription factor CMAF is important. So TGF-beta is critical, so is IL-6 for the differentiation and function of Th17 cells. Now, what is important is that there are at least two distinct isoforms of TGF-beta, which can lead to a little different in terms of the outcome. But more importantly, TGF-beta signaling, if it is missing, we do not see a Th17 cell. IL-6 signaling is very important because IL-6 is going to activate the STAT3 molecule, and IL-6 STAT3 molecule is absolutely important to downregulate the FOXP3, which is going to get activated in the presence of TGF-beta only. So FOXP3 induced by TGF-beta is able to suppress Th17 cells, and they do that by antagonizing the ROR gamma T as well as ROR alpha from the Th17 cells. Additionally, interleukin-23, since the receptor of interleukin-23 is not expressed by the naive T cells but by activated T helper cells, IL-23 has been shown to be important to maintain the phenotype of Th17 cells. And IL-23 is very important to drive GM-CSF expression from Th17 cells, and that leads to the pathogenic property of Th17 cells. The signaling of interleukin-21 can replace IL-6, and it can always act as an alternative cytokine in vitro. IL-21, if it is working with TGF-beta, it can induce the expression of ROR gamma T in the absence of interleukin-6 molecule. And so if we just summarize some of the key players in the signaling of T helper 17 cells, downstream of TGF-beta, we could see SMAD2, and SMAD2 can interact with tripartite motif TRIM33 to induce the regulation of Th17 cells. So TRIM33 it is a modulator of TGF-beta signaling. It associates with SMAD2 to regulate the pro-inflammatory function of interleukin-17. TRIM33 can induce Th17 but not T8, so that is very important. So it's a tripartite motif containing 33. So that is one of the motifs we can see here. Downstream of interleukin-6 STAT3 signal, there is another motif which is TRIM28. So IL-6 STAT3 helps in TRIM28 binding to the interleukin-17 locus, and TRIM28 actually forms a complex with STAT3 and ROR gamma T, which usually regulates the super enhancers triggering the Th17 cell differentiation. And downstream of TCR's CD828 signaling, we can see IRF4 and BATF, which would actually open up the ROR gamma T. But this is very important because this IRF4 and ROR gamma T, BATF, these are also the transcription factors which are important for the regulation of Th9 cells as well. So we could see that some of these transcription factors are shared between Th9 and Th17 cells interaction. So IRF4 can work with transcription factors like STAT3 and BATF to regulate the expression of interleukin-17 gene. And BATF can actually open up the chromatin during Th17 cell differentiation. So it can facilitate the binding of IRF4 to the DNA, and it generally, BATF interacts with IRF4 to induce the production of interleukin-17 and interleukin-21 in Th17 cells. So overall, we could see a very complex network of Th17 cells as well. And then complexity gets increased not only because of the various cytokines which are required for the differentiation of Th17 cells, but also what those cytokines, what those T helper cells, 17 cells are also secreting. So if it is secreting interleukin-17, where they are acting, if it is secreting interleukin-22, who are the activators of interleukin-22. So from naive T cells, which is CD3 positive, CD4 positive T helper 0 cells, Th0 cells, in the presence of these polarizing cytokines, we could see a differentiation of T helper 17 cells, which could secrete various cytokines. In helping the immune responses, Th17 cells are very important to fight off any pathogens at the barrier or at the mucosa. They also recruit, because of the release of these cytokines and GM-CSF, they can recruit some of the neutrophils to the site of infection in order to perform in order to expel any potential parasites or any potential pathogens, extracellular pathogens, which are coming down those barriers. So the release of antimicrobial peptides helping in the barrier function, releasing certain chemokines and cytokines to help in this process are the good roles of Th17 cells. But like Th9 cells, which if not regulated, Th17 cells, it is very well known to this audience, has been shown to be involved in various autoimmune disorders. And that comes because of the categorization of the T helper cells into the pathogenic version and the non-pathogenic version. So TGF-beta 3 isoform with interleukin-6 gives rise to pathogenic T helper 17 cells. But if the TGF-beta 1 isoform is used in concert with interleukin-6, we can see that not only these cells are secreting interleukin 17, interleukin 21, but they are also releasing interleukin 10. And for the releasing of interleukin 10 gene, there are additional transcription factors which are responsible, mainly CMAF, AHR, and the transcription factor ionos, which also comes to the Helios family of members. The non-pathogenic T helper 17 cells also express certain specific molecules like the CD5L. It also increases the expression of some of the ectopeptides like CD39, CD73. And if these cells are generated, somehow these cells can stop the progression of inflammatory bowel disease, can somehow stop the progression of rheumatoid arthritis. If these cells are going to be transferred to in a mouse model to induce type 1 diabetes or EAE, compared to wild type or the pathogenic T helper 17 cells, these non-pathogenic T helper 17 cells which are expressing interleukin 10, they fail to do so in an efficient way as their pathogenic counterparts are actually doing. And like the Th9 cells, which we had discussed having both pro-tumor and anti-tumor properties, Th17 cells also have these properties as well. So the pro-tumoral Th17 cell properties are related because of the promotion of angiogenic molecules and the recruitment of myeloid-derived suppressor cells. The plasticity has been discussed in this forum, so I'm not going to talk about it, but T helper cell 17 cell plasticity, T helper 9 cell plasticity, they are also going to be relevant in either generating an immunosuppressive environment or promoting an anti-tumor pro-inflammatory environment. The anti-tumoral T helper cell properties will include the recruitment of some of the cytotoxic T cells, CD8 T cells mostly, and also stopping or recruitment of the dendritic cells, which could actually present the tumor antigens to the CD8 T cells in a better way. So depending on what tumors we are looking at, what cytokines are present, and what other molecules are expressed, T helper cell 17 cells can also have either a pro-tumor activity or an anti-tumor activity. And naturally, interleukin 17 gene, since it is one of the key genes which is secreted by Th17 cells, has been targeted for different diseases, mostly the autoimmune diseases. So there have been targets to the interleukin 17H family, interleukin 17F family member, the heterodimer of interleukin 17A or 17F. The receptors, they have also been targeted with different antibodies, monoclonal antibodies, which came with different results. So I have just summarized some of these antibodies which are shown here. So tocilizumab is a humanized interleukin 6 receptor specific antibody, and it has been approved by FDA for rheumatoid arthritis or systemic juvenile idiopathic arthritis. Cetuximab is also a IL-6 specific monoclonal antibody, but it is a chimeric IL-6 specific monoclonal antibody which is targeting the Castleman disease. The interleukin 23, P40 and P19 subunits have also been targeted, and some of the biologics have been approved for either the psoriasis, psoriatic arthritis, or plaque psoriasis. And the last two, secukinumab and ixekizumab, they are basically humanized monoclonal antibody which are targeting interleukin 17A and have been approved for various diseases. What is important is that even though I have mentioned that Th17 cells are responsible for inflammatory bowel disease, but targeting interleukin 17 has not been shown to be beneficial to target inflammatory bowel disease in some of the studies. So secukinumab actually had no effect on IBD, which can be attributed to the functional transformation of the Th17 cells in the background of different diseases. So what we need to think in the broader perspective is how to select these key players which are specific in autoimmune diseases, or what kind of cytokine circuits are critical to be targeted for targeting a particular disease. We also need to appreciate that there would be some redundancy in the action of various inflammatory cytokines, and that might limit the efficacy of a single neutralizing agent. So we have to be a little bit more careful in terms of doing or creating an antibody against a particular cytokine. Some of the studies which were initially done in mouse, they do not necessarily translate into the human studies. So we need to get innovative translational studies on human patients, specifically with respect to cancer. So that is very critical. So even though if we are targeting the cytokines or the cytokine receptors, we also need to understand that these guys are also important to fight the host against various pathogens. So a broad targeting of a particular cytokine or a cytokine receptor might need to be verified for this purpose. And blocking the cytokines to prevent, therefore, could lead to the susceptibility of various infections. Or maybe there could be a risk if the cytokine was helping in an anti-tumor response, we could see a break in that chain of anti-tumor immune responses. So everything has to be, we need to appreciate why a particular cytokine is, if it is targeted, why it is not giving the desired output. For example, interleukin-17, the clinical success has been also fairly limited in rheumatoid arthritis, and I'm not going to talk about this because I guess I'm fairly out of time. So with that, I would like to again thank the society for giving me this talk, and I'm open to have any questions, answers at this point.
Thank you, Dr. Goswami, for the enlightening talk on the importance of Th19 and Th17 in various diseases. Now this talk is open for discussion.
Comment. The Tocilca, the interleukin-6 inhibitor, is used in the uveitis with cystoid macular edema quite successfully. And this is one of the interleukin-6 inhibitors is found to be very efficacious.
Thank you, sir.
So, Rito, it's a fantastic talk and very good information about Th9 and Th17 cells and how they contribute in different diseases. Just for the sake of broader audience, I just want you to just discuss in few words that the positive side of the Th17 cells, especially given its antifungal nature and the current scenario.
So I think, can I just say that again? I couldn't hear you clearly.
So like Th17 cells actually known to have antifungal activity.
Right.
And now we know in COVID time that the post-COVID maintenance, the fungus is one of the, especially with this high steroid treatment.
Right.
So having a lot of this immunosuppression and also losing these Th17s, how they talk to each other in terms of infection and Th17.
Right. I mean, so, and that is something I have mentioned at the end, actually. Like if we are targeting interleukin-17 per se, we tend to lose its potent function against the pathogens. And so if we are looking to target any specific cytokine, we need to really rationalize why that cytokine is only targeted and what are the players around that environment. So ad hoc clearance of interleukin-17 just to maybe stop the inflammation is probably one part of the story. But with respect to COVID-19, I think Amit can even enlighten us even more. It's whether the mucormycosis, the stage of that disease, when that mucormycosis is actually taking place during COVID-19, whether the role of Th17 cells is important at some point of COVID-19, is whether interleukin-17 is actually helping, because in some of the diseases, interleukin-17 is helping when the disease is, or actually making the disease worse when the disease has already started. In some diseases, it is probably helping to start of the disease. For COVID-19, we need to understand where this interleukin-17 might have this antifungal stated, and it's complicated. I think you can probably share some ideas. I don't know what you are thinking, but to just a broad clearing of a particular cytokine is probably not always the answer, other than interleukin-17. So I think that might be one of the things, and I think the receptor signaling is also critical, because the receptor probably, if you're targeting a cytokine which is going systemically through the body, versus the receptor that is going to be expressed by certain cells. So I think targeting those receptors might be important if we are actually trying to downregulate a particular effect of a cytokine. That is my personal thought.
Right. Thanks, Rito. I think it's a complex biology, and very, very complex biology, that at what time and what location we need to intervene. Even if it is Th17, which tagged as pathogenic sometime, non-pathogenic on sometime.
Right.
So thank you very much. Thank you.
Thank you. I think the one thing I just want to mention very briefly is like maybe to think about the epigenetic modifications, the epigenetic drugs, and some of these things are shown to be very efficient because they are controlling some of the super enhancers. So the BET domain has been targeted by the pan-BET inhibitor JQ1. It's shown some really promising. And the other thing which I really didn't have time to think is how about microRNA as a therapeutics, which are either directly targeting cytokines or indirectly targeting the cytokines.
Perfect. Thank you.
Great. Thank you.